I always like to start off a lecture, with a pretty picture, but this pretty picture bears importantly on our discussion today about major evolutionary transitions. And so everything that's visible here is multicellular, and of course each one of these organisms has associated with it, many more microorganisms than actually the cells in each of these bodies. I'm not going to talk about those so much today as I am the major transition that occurred when unicellular forms evolved into multicellular ones. So, without further ado, this is our title for today, Dissecting Major Evolutionary Transitions. And this is just a brief outline. I'm going to give you a history of this project, as well as the history of the idea itself of major evolutionary transitions, acronym MET. I will focus on one of these. There are several, from single cells to multicellular organisms, and discuss a little bit about how might we study the evolution of multicellularity. Clearly, something which is an important driver of complex life here on Earth. And then the data part of the talk will consist of insights that we've gained from two complementary approaches, that of experimental evolutionary genomics and comparative genomics. And we'll finish up with a future directions and then acknowledgements and how I see this kind of work as being relevant to the IOE going forward. So this project, Experimental Evolution of Major Transitions, had its origin as a NASA Astrobiology Institute award, which was awarded to the U of M in 2015. Involved these people here, some of whom went with me to Georgia Tech, and others of whom remained back at U of M or were at other institutions. And so this was the NAI grant, and the NAI had a proud history for a number of years, but in fact, the directorate at NASA decided to sunset this project beginning back in 2020. So this was the history of the NASA Astrobiology Institute, reaching over about 23 years. And in its place now, and this actually bears importantly about, the future of work like this and the IOE, is that NASA has elected to go with something called these research coordination networks. And the distribution of these 5 RCNs was in keeping with the 2015 NASA Astrobiology Strategy document, and it involves these 5. These 5 have now come all into existence. The first was the Nexus RCN, which was dedicated to exoplanet study and exoplanet habitability. Then in 2018, the Network for Life Detection. And then in 2019, the Network for Ocean Worlds and the Prebiotic Chemistry and Early Earth Environments RCNs came into existence. And just a couple of months ago, the last of these, I wouldn't say it's come online. It will be launched in May at AbSciCon, but it is tentatively titled From Early Cells to Multicellularity. And this RCN, of which I'm a part, is dedicated to advancing studies that range from the early evolution of metabolism to the advent of multicellularity, which, as I'll show you, has occurred across the tree of life. Now, whereas the NAI came with a chunk of money, in most cases in the order of $7 to $9 million, the RCNs are individual projects are funded separately by different programs within planetary sciences at NASA. Just want to make that clear, that I am, that Ariel and Mary and Bektul and I are not getting a pile of money to do this. This is really a community grassroots activity. All right, so back to the main event. major evolutionary transitions. So what is a major evolutionary transition? It's really when you're going from simple subunits to more complex wholes, where typically those more complex wholes are able to store and transmit information in a qualitatively new way. An important feature of major evolutionary transitions is that these more complex wholes, the subunits that make them up come to be interdependent, and this new unit has a degree of autonomy. Once that's been established, that makes possible the division of labor amongst subunits, and for sure, to the extent that this new entity becomes the object of natural selection, then it is the unit of adaptation, as opposed to, say, this single cell over here. I'm going to put on my little laser pointer here for a sec. Okay, we took this concept from an exceedingly influential book by John Maynard Smith and Eeyore Zothmary that was published in the late 90s. And they envisioned a number of such transitions, including that from RNA to, to the two polymer system of DNA, nucleic acids and proteins, the transition from prokaryotes to eukaryotes, the invention of sex, and of course, the transition from unicellular life to multicellular organisms, the evolution of multicellularity. And so we're going to focus on this particular major evolutionary transition, and I'll show you how this project that really originated in my laboratory with the advent of a really great postdoc and scientist, Matt Herron, has morphed into something that's really kind of, I wouldn't say it's taken over, but it's become a very important part of our research portfolio. Okay, so from early cells to multicellularity, these major evolutionary transitions have been driven by cooperative interactions. So the little circles that I showed on the prior slide, when you get this more complex circle consisting of these 4 subunits, the implication is that they are interdependent and cooperating in some manner. It may be that competition against other such units or as single cells drove this evolutionary transition, but once it's established, a certain amount of cooperation is required. And so I view major evolutionary transitions as including things like the acquisition and elaboration of organelles, also the many different types of symbioses that exist across the tree of life, and some of those symbioses including syntrophy in the microbial world, and of course the origin of multicellularity. And there's multicellularity within bacteria, obviously with certain cyanobacteria as a classic example of multicellular organisms that have differentiation with heterocysts and somatics and vegetative cells. And of course, across the eukaryotes, there are many, many instances of multicellularity. So here you have 2 simple multicellular organisms. On the bottom right, you have Volvox carteri. This is this freshwater chlorophyte alga that has fascinated biologists since Leeuwenhoek and Linnaeus. And here's a sponge up here on the top left. The presumptive ancestral types of organisms that gave rise to these are, in the case of sponges that consist of these choanocyte cells, choanoflagellates, supposedly the great granddaddy of animals, bearing in mind that current choanoflagellates have their own evolutionary history that has been pursued independently of animals, for several 100 million years. Likewise, it is reasonable to postulate that an organism like Chlamydomonas, a single-cell chlorophyte alga, bona fide eukaryote, is that a form like that was the ancestor of the current Volvox colonial form. And just like this organism over here, Volvox has full germ soma differentiation. And furthermore, you have amongst the gametic cells that you see large in the center of the Volvox colony, you have small motile sperm and very large, in the case of Carteri, immobile eggs, so oogamy, and we'll raise that term later on. So how do unicellular forms evolve into multicellular ones? In other words, how did multicellularity arise? And after it did, how and when did cellular differentiation and various forms of sexuality follow? Because once you get an unequal investment by parents in eggs versus sperm, once you have anisogamy, a big difference in the size and content and the expense of making these, then all other forms of sexual differentiation and sexual selection follow. Well, it turns out that multicellularity has independently arisen at least 25 times in the eukaryote. This is an important point because it implies that in these different lineages, there may be different genes that are involved in mediating this major evolutionary transition. It may also be the case that there are certain genes in common across all of these independent evolutions that prove to be important, but we don't really know that yet. One thing to note in this slide from Strathman and Grossberg a number of years ago is that you have some clades where all members are multicellular. So, for example, ourselves, the animals, we are all multicellular. So also the red alga. So also plants and slime molds. But there are other groups like the chlorophyte algae. This includes the volvocine algae, where you have some unicellular representatives and you have some colonial multicellular representatives. And that turns out to be really important for everything else we talk about this afternoon. So how can we study the evolution of multicellularity? Well, one thing that we can do is study the fossil record. And this has been useful in many cases. And so, for example, it's because of this fossil, Bangiomorpha pubescens, that we believe that the red algae evolved multicellularity at least 1.2 billion years ago. So that's great. But if you think the fossil record is relatively poor with respect to large multicellular vertebrates, it's even more sparse with respect to small organisms like this, and even more so organisms that are in freshwater habitats. So this is great. Microfossils are wonderful. They're necessary for doing molecular clock calibrations, but you have a fairly sparse record of the origins of multicellularity and the early evolution of multicellular organisms. Another approach, a comparative approach, chiefly based on morphology and cell biology, is to compare multicellular forms to extant unicellular relatives. And so, as I said before, this choanoflagellate is or an ancestor like this is supposedly the ancestor of animals, including organisms like sponges, again, clammy and volvox, and yeast-like organisms and these mushrooms. Now, one problem with this approach is, of course, that these extant unicellular forms, as I said before, have had their own evolutionary trajectory over the same amount of time as these multicellular forms that are relatively advanced that you see on these bottom panels. So that's an important limitation of that approach. Now, sort of a variation on this which has proved to be really interesting in a number of different clades, is we can compare members of a group that vary in cell number and degree of differentiation. So in that Aries slide from Grossberg and Strathman, I pointed out that there were some clades, like in the chlorophyte algae, where you had groups that had both unicellular and multicellular forms. And this is true in the freshwater volvocine algae, which include things that are single cell, like Vitrio chlamys and Chlamydomonas, and these complex multicellular colonial forms that are in the genus Volvox. Importantly, within this group, which is a fairly closely related, phylogenetically speaking, and of a reasonably uniform size in terms of their genomes, are these intermediate forms where the characteristic cell number is 4, or maybe 16, or maybe 8, as well as these others, and with varying degrees of cellular differentiation, from no cellular differentiation but multicellular to full-on differentiation into German soma, as we see in the advanced Volvox species. So this is kind of rich territory for doing comparative biology, giving rise to ideas like this, adapted from an essay by David Kirk back in 2005, where he did something kind of akin to what Darwin did with the vertebrate and the cephalopod eye. He tried to imagine the discrete steps that would take you from an ancestor that was similar to Chlamydomonas, being unicellular, all the way up to this complex form of Volvox, like Volvox carteri. And he came up with this 12-step program, starting with incomplete cytokinesis, all the way up to complete differentiation and a bifurcated cell division program. So this comparative approach in certain groups of organisms has proved to be very fruitful. Now, another approach, and this is really originated with Will Ratcliffe and Matt when they became associated with me, or me with them, is we can try to evolve something in the laboratory that starts to resemble something like Volvox, but beginning with one of the unicellular forms that we have at our disposal. And we did that. And so now I'll give you a few slides of insights from experimental evolution. And I'm kind of cherry picking from a number of papers that have come out over the last 7 or 8 years. But before we do that, let's imagine some advantages and disadvantages to multicellularity. I think many of us contemplating this think, well, what's the disadvantage to being multicellular? And I'll show you that there are actually some problems. So there's multicellular organisms are obviously larger, and that gives them squatters' rights, potentially to scarce or limiting nutrients in the environment. That gives them potentially protection, both from biotic challenges like predation, as well as abiotic challenges in the sense that a larger body may be able to better buffer environmental variation. And then, of course, once you have modules, remember that slide with the 4 circles bounded by the dotted line. Once you have modules, then you have an entity that's open to division of labor and the presumed efficiency that comes with that development. But there are disadvantages. So yes, large size solves some problems, but it creates others with respect to the surface area to volume ratio, the former, the square, the latter, the denominator here, the cube. This creates problems for diffusion and transport. And so organisms that become large multicellular organisms have to invent some sort of a circulatory system or open themselves up internally to the external environment in order to bring nutrients in and to let wastes diffuse out. Another disadvantage to being multicellular is something that I'll call genetic conflict. Now, this arises when one module pursues its own interest at the expense of others. And you might think, well, this doesn't make any sense. Well, maybe it doesn't. But in fact, mutation, somatic mutations can produce this. And I would put it to you that, that cancer is an excellent example of genetic conflict in our own bodies, where we really have cells that de-differentiate and take off on their own evolutionary trajectory, often, ultimately, at the expense of the whole organism. All right, so large size. That should be a way that we can select in the laboratory for multicellularity. So Will and Matt did some very simple experiments using centrifugation to select for large size in Chlamydomonas reinhardtii, a unicellular organism. This is the way it goes. A short, slow spin in a microfuge tube. You throw the top 90% into the trash, and you transfer the rest to fresh medium. The reasoning being that those larger particles in the microfuge tube will settle more readily under these conditions than will those that are only single cells. And so they did this for several hundred generations, and they did 10 without settling selection, 10 with settling selection, never got multicellularity in the absence of settling selection, and in 1 out of 10 replicate populations, they got this startling result that was published some years back in Nature Communications. They evolved multicellular Chlamydomonas that had their own life cycle. So here you have this large cluster that will disperse by the single cell propagules, and these single cell propagules will go on to found their own colonies. And basically what you see are these clusters that grow clonally from a single cell ancestor. So here we have examples of going from this phase, from 1 or 4 cells to this larger entity. And this is heritable in the sense that you can put these away in the refrigerator and bring them back out and repeat this this type of life cycle. So this was an interesting report that got a lot of attention a number of years ago. Ratcliffe and Heron, really a lovely piece of work. But what might be a more ecologically relevant way to select for large size in unicellular Chlamydomonas? Predation. So for those of you who have dipped your toes in the protist water, very often if you ordered paramecium from Carolina Biologicals, they'll send you a culture, and that culture is usually being fed with Chlamydomonas. And in fact, here you have paramecium that is engulfed a number of clammy cells. And so if you think about it, a large multicellular clammy would have an advantage with respect to the unicellular forms in terms of avoiding predation by this particular protist predator. So Kimberly Chen and Jacob Boswell, a postdoc and an undergrad in my lab when I was still at U of M, came with me to Georgia Tech. They embarked on this enormous experiment where instead of doing centrifugation, they did much slower growth and much longer transfer times in the presence or the absence of predators. And so here you have their simple experimental design. They crossed a couple of clammy strains in order to get genetic variation in their populations, and then established replicate populations, 8 of them in total, 3 controls in the absence of paramecium tetraaurelia, and 5 that were cultured in the presence of the of the paramecium and transferred on a weekly basis. So this experiment went on for a whole year, after which we started to analyze the populations that are described here as B1 through B5, K1 through K3. In a subset of these populations, in none of the controls, but in a couple in the B2 and B5 populations, we saw forms like this evolve. And these, again, were stably heritable forms. Now we see something completely different. Not these kind of amorphous clusters, but these forms that are encased within this common cell wall and have a characteristic cell number. So here they are. Those I just transposed the pictures that were off to the side and put them on the top half of this. And this is evolution of multicellularity in response to predation. But on the bottom are three bona fide volvocine algal species, Pandorina volvulina and Yamagashii. And I would put it to you that there is a remarkable resemblance between these forms that were evolved in the laboratory, de novo multicellularity on top, and here you have these forms on the bottom, which are bona fide volvocine algal species. I am not going to claim that we have forced speciation as well as evolved multicellularity, but it is a striking result. And it is also the case, as I've said a couple of times, that what you observe here on top are stably heritable forms with a characteristic cell number. So this paper was published a couple of years back in Scientific Reports, where it was, I think, one of the top 5 cited papers that year. So what Will and Matt and Jacob and Kim found was that we evolved in these different populations, different life cycles, right? So if you look down here, so here are these 4 different life cycles where you have, in some cases, single cell propagules. In some cases, you don't get the evolution of multicellularity, even in cell lines or cell populations that did give rise to multicellularity. There's a mix in there. But you have some forms that have single cell propagules, others that have that essentially reproduce by a kind of fragmentation. But in all cases, what develops, whether it's from a single cell or whether it's from a characteristically sized fragment, the development is clonal, meaning that you, in fact, have a true multicellular life cycle. So just to reinforce this point, the propagule sizes vary amongst these isolates. And so looking at a 72-hour time-lapse video, and taking large numbers of images of strains, we see on the bottom, this is log 2. So these are single cell propagules. And up here are particular isolates from these populations evolved in the presence of predators that had multi-celled propagules. So what do these things look like in a time-lapse video? Well, here's one. So these are starting from single cells, and you'll see in the population going up to tiny multi-cells. So this is more like what you would see in a control, because in the process of first undergoing cell division, Chlamydomonas will transiently have 2 to 4 cells present in the field of view. So here's a multicellular form that is growing and giving rise to other multicellular forms. And here is another one, the same sort of image that you saw in the previous time-lapse video. And again, from isolate to isolate, these time-lapse videos yield a characteristic life cycle as depicted in that figure that summarizes the life cycles that we saw, these panels A through D. So we evolve in some, but not all, of these populations, heritable multicellularity. Does this provide any sort of selective advantage? And so, so Matt and Jacob came up with a cool way of analyzing this, and that is simply to compare the absorbance over time in populations that either did or did not have a predator, and subtract those two. So you would have 4 replicates multicellular, the same 4 cultures, but without a predator, with a predator, same with unis, with and without predators. And you can see here that if you the change in absorbance measured at 420, if you have predation occurring, you have less clammy dispersed within the medium, and it's this change in absorbance that is essentially the way that we're scoring the intensity of predation over time. So here you have multicellular in the open diamond and in the black square unicellular, and here's the whole enchilada of all of these experiments. And there's a pretty, and again, bear in mind that 0 means no change. So no change in these multicellular forms, but a pronounced difference between with and without predator in the unicellular forms. And if you take it all out, to the very last point, then you do an ANOVA on this and see that the phenotype of either multicellular or unicellular has a strong effect on algal density. So the takeaway from this is that these unicellular forms are much more susceptible to predation than the multicellular forms, suggesting that the predation pressure was why we have the evolution of this phenotype. So we always want to know what's the genetic basis of this evolved multicellularity. And I'll give you some preliminary results. This is Kim Chen's work, and she is in the process of analyzing these data, but I'll tell you what she's up to. So here are the controls. Here are the strains, the populations in which we observe some multicellularity. And so she's analyzing individual isolates from B2, from B5, these are predator evolved, and K1, these are evolved in the absence of predator, but starting with the same population, and is doing paired end Illumina sequencing with the objective of uncovering mutations that are in multicellular forms that aren't in the unicellular ones. So in this diagram, the rows are isolates from one of these evolved populations. So here are 4 independent isolates from the B2 population where we saw some instances of multicellularity. Same deal here, B5, and here from the K1 population are 4 independent isolates. In the columns are chromosomes. So this column is, let me get my laser thing going here. This column represents chromosome 1, this column chromosome 6, this one chromosome 10, and there's the in 17, chromosome 17. And what she observed is that there are shared mutations that are shared amongst the multicellular isolates in B2, but a different set of shared mutations that are shared across the B5 population. And recall that these gave rise to qualitatively different forms of multicellularity with respect to how large they grew, as well as their propagule size. So there appears to be 2 genetic ways to sort of skin the cat. So she wants to get beyond this to actually the causal mutations. And for this, she's taking a bulk segregant analysis approach, doing essentially meiotic purification by crossing multicellular forms with ancestral unicellular forms, generating in the process, oops, why are we not able to advance? Generating a bunch of recombinants and then selecting on those recombinants so that she's selecting the unicellular forms and enriching for those genes or those intergenic regions that are associated more with unicellular forms, and selecting on those that are multicellular to try to purify for those loci and or intergenic regions that are associated with multicellularity. And so this is just another way of viewing the same thing. She's using settling selection to distinguish between these, and she did 4 rounds of these settling selection experiments, enriching for multicellular forms versus unicellular forms. And then these become the pools for doing bulk segregant analysis. What has she observed so far? Well, in this particular isolate from the B2 population, we see de novo mutations enriched on 3 of the chromosomes, chromosomes 4, 11, and 13. And the bulk segregant analysis also enriches for regions of the ancestral genome that are associated with multicellularity. I'm not saying that these control multicellularity, but they may have come under the control of mutated regulators, and it's that altered regulation that makes them show up in these BSA analyses. So here's the B2s. Here is an isolate from the B5 population that had stably heritable multicellularity. And you see, again, you have the enrichment of de novo mutations as well as certain ancestral regions that are carried along in the B5 evolution of multicellularity. The important thing to note is that going from B2 to B5, as we saw with those chromosome, those earlier pictures before she did the BSA, is that these 2 different cell lines evolved different forms of multicellularity with different life cycles through different genetic means. So you can have heritable multicellularity arise by different mechanisms. And we shouldn't be really surprised at that, in the sense that we know that multicellularity, unlike, say, the symbiosis and the major transition that occurred when the eukaryotic lineage, the lineage that led to the eukaryotes, acquired a mitochondrion-like ancestor and gave rise to that particular evolutionary trajectory. Multicellularity has arisen across the tree of life, and even within closely related organisms by apparently different mechanisms. So we've had these 2 different forms of multicellularity: settling selection on top, predation selection on the bottom. Have we achieved something that begins to look like Volvox? Well, maybe in the bottom, not so much at the top, but the fact that you have heritable multicellularity and that is underpinned by these genetic changes suggests that we are at least on our way, and it's going to require a lot of creative experimental evolution going forward in order to learn more. So what we've seen is that experimental evolution can select for 2 of Kirk's steps. In both of these multicellular entities, not going to call them new species, but they're certainly organisms that we have selected for extracellular matrix. So we show in that earlier 2013 Nature Genetics paper how you have extracellular matrix built up within this mass. You also have extracellular matrix derived from the cell wall in the predation selected multicellular lines. But you also have in predation selection one of these other key features, which is number 6 on Kirk's 12-step program, the genetic control of cell number, because the non-reproductives in the case of these more recent experiments do have characteristic cell numbers. Okay, So we can get multicellularity under selection in relatively few generations. In the case of the recent experiments, we show the reduced risk of predation. These are new units of adaptation. We have some genetic understanding that's now within our grasp. But a point to emphasize is that evolution of multicellularity is not inevitable. That is to say that in two of 5 selection lines in the predation experiments, we didn't see any multicellularity. And in only 1 of 10 settling selection lines did we see the evolution of multicellularity. And even within those predation selection lines where we saw this sort of result, there were instances where we did not. There were other organisms that were with them, genetically different, that had remained unicellular. Okay, so in the next 10 minutes, I'm going to give you a quick tour of some more recent work, insights from comparative genomics. And so here's a paper by Matt. This came out before he joined my laboratory when he was still a grad student at University of Arizona with Rick Michaud. And he did this wonderful phylogeny that was molecular clock calibrated of the volvocine algae. And in that paper, he challenged and disproved the earlier hypothesis that this group of organisms evolved about 50 million years ago. His analyses suggest that they evolved between 200 and 250 million years ago. So this is an evolutionary hypothesis, as are all phylogenies. And in this case, Matt's phylogeny was based on really just a couple of, actually a handful of chloroplast genes. Now take a look at some of these species that I have pointed to here on the left-hand side. This view, this phylogeny, which is a solid one, albeit with few genes and few sequences within them, invites a progressive view of the evolution of complex life. So here's this paper by Jim Uman a couple of years ago, where he takes those species in the arrow that I just showed you and maps them onto this 12-step program, but now with a time axis on the bottom. And here's clammy, current day clammy, and here's current Volvox carteri, going from unicellular to colonial multicellular. Well, these views and attempts to reconstruct the ancestral states, including these 12 steps, should be predicated on a true phylogeny. But if you take stock of the phylogenies that have been produced to date, and here you have gray as the origin of cell differentiation, colors as being the different genera in these different analyses, what you see is a pretty mixed bag. First of all, this is purely on phylogeny. Here you have a couple of internal transcribed spacers, from the 18S ribosomal subunit. Here's chloroplast DNA. So these are taxon, numbers of taxa relatively few, and the number of genes even fewer. And perhaps it's not surprising that these phylogenies are not concordant. So this student of mine, Charles Lindsay, just published a paper this fall in BMC Biology. It was one of the 2 papers of 21 to be in the Light of Evolution series. So really proud of this young man who did a fabulous job where he did gene expression analyses, collected the whole transcriptome on 55 algal strains, and then went to the literature and got information on 13 more. So really increased taxon sampling, looking at 68 different strains, and then sifted through that expression data and came up with 40 orthologous single copy protein coding nuclear genes, not this chloroplast business, and analyzed their phylogeny using different phylogenetic inferences. So these inferences are based on a 40 protein concatenated data set, about 2,600 aligned amino acids. And so among his conclusions in this paper are, first, the bulbocene algae are not monophyletic. You've had, even within this group, you've had at least two independent origins of multicellularity. You've had an independent origin of multicellularity within this group that includes Chlamydomonas and Vitreochlamys, but also Tetrabena socialis. And you've had another evolution of multicellularity in this group that gave rise to these organisms here. Something else that he found is that somatic cellular differentiation has independently evolved at least 4 times in this group. Here in the Volvoxes, here in Astrephamine, as well as in this group giving rise to other Volvox groups and Pleodorama. It also turns out that this trait, isogamy, is where you have gametes of equal size. So all of these organisms have a sexual phase. They undergo meiosis and produce gametes. The primitive phase, and this is what you see in Chlamydomonas, is isogamy, where you have plus and minus cells, and these different gamete types come together to have a plus minus diploid zygote, but they are the same size. However, anisogamy has independently evolved at least a couple of times in this group. Let me silence this and go back to automatic. Try to get rid of this thing. Sorry about that, guys. That anisogamy has independently evolved at least 3 times. And this is only possible by doing this massive analysis. Anyway, so some important insights gained by doing comparative genomics. All right, in the last few minutes, I'll tell you future directions. So Ross is in the process of working with Andy Knoll at Harvard and building up a chronogram for the origins of multicellularity, differentiation, and the sexes. And also, the ultimate objective is to do ancestral state reconstruction with a much improved phylogeny that is also calibrated in the fossil record. There have been a number of new fossils that have come to light in across the plant kingdom that can be used to inform and better calibrate this than the time tree that Matt produced back in 2009. So as far as experimental evolutionary genomics, we would like to select for motile multicellularity all of the forms that even those pretty spheroid forms with the characteristic cell number. One trick that they haven't performed is everting their flagellae. So whereas forms like gonium and Pleodorina are fully motile and do phototaxis, chemotaxis, geotaxis, etc., these organisms cannot. They escape from predation by size, but they cannot escape from potential predators by swimming from them. So it may be that, at least as far as we have taken them, they're kind of an evolutionary dead end. We would like to select for differentiated multicellularity, and we would like to see if we could select for anisogamy, and another postdoc, Pedram Samani, has made some pretty amazing progress along those lines. As far as functional genomics of major transitions, surprisingly, no one has done or capitalized on single cell omics in looking at say RNA-seq differences, expression differences in the cells that are either somatic cells or gametic cells in Volvox and more primitive forms of the bulbocene algae. And I think that's a potentially very fruitful area of investigation in the future. Okay, so acknowledgements here in the last minute. Here's Matt Heron, great postdoc. He's now a program officer at the National Science Foundation. You guys, some of you may interact with him on a regular basis now. Will Ratcliffe is now an associate professor here at Georgia Tech. These are just terrific young scientists, and it's been really one of the great blessings of my scientific life to have had them in it. And then Jake Boswell, Chuck Knox, Margaret Boyd. These were undergraduates with me at the University of Montana. Margaret's now at Northwestern, wrapping up her PhD. Josh is at UC San Diego now, wrapping up his postdoc, and everybody else has been associated with me here at Georgia Tech. And of course, the people paying the bills, a little bit from the NIH, but most of it from NASA. Now I see that I've gone 51 minutes, which is a pretty good clip. And I had one more slide, and that is to let you guys know what I think the relevance of a project like this is to the IOE. And then hopefully we can have some discussion both about the science and about whether you agree or disagree with this slide. First of all, I think that certainly at the University of Montana, ecological and evolutionary genomics, whether it's basic or applied, should become a cornerstone of the U of M IOE and therefore the Montana IOE. I would put it also to you that studies focused on compartmentalization and division of labor are particularly relevant to folks who are doing ecosystem science, in the sense that any sort of modeling, compartment modeling, that would be applicable to one, the same general principles should be applicable to the other. And in fact, I'll just let you know that I'm really interested in this theme of compartmentalization across scales actually from doing channeling of substrates and multi-enzyme complexes. Those are arguably compartments, up to symbioses, to centrifuges, to multicellularity, and to the partitioning of function within ecosystems. I'd also say that this new NASA RCN, early cells to multicellularity, is a natural fit for a lot of folks on both campuses. And concerning not just that RCN, but all the other RCNs, because there's plenty of people at MSU in particular who would be candidates for membership if they're not already members of the PCE3 group, is that they were established with the objective of cross-agency funding, particularly between NSF and NASA, but also between other federal agencies and NASA. And in fact, these RCNs were born of an ideas lab that was run jointly by NSF and NSF and NASA about 4 or 5 years ago. Anyway, that's it for me, folks. I really appreciate your attention. I look forward to hearing some questions. I'm also looking forward to seeing you guys in person, hopefully sooner than later. At least see you this summer, if not sometime later on this spring. So I thank you for your attention, and that's it.
Thank you so much, Frank. That was a really interesting presentation. I think we have a small enough group that if you'd like to turn on your videos and just popcorn questions, I think that would be an appropriate way to move forward.
I'm trying to, there we go. All right, so let's see. I want to expand this panel to show who's here. All right, so it's the few and the proud.
So Frank, Tim here.
Yes,
yeah.
Sir.
So can you guys manipulate this? This sort of goes back to your previous life when you were messing around with when you were Pseudomonas aeruginosa jock and biofilms.
Yeah.
biofilm matrices, etc. Can you guys manipulate the multicellular phenotype by, say, a macronutrient starvation, for example? I mean, so where this derives from, this question is like a lot of gram negatives, their extracellular polysaccharide formation is controlled by genes that light up, that are expressed highly under phosphate starvation conditions.
Yeah.
Okay. And so it's those kinds of things that, immediately come to my mind, that there's a surface entity that's being overexpressed, that makes cells sticky, and they start clomping onto each other one way or the other. And so that's the first question. The second question, what genes are being mutated?
Well, the question number one, we certainly could manipulate, for example, so you were suggesting, based on what's known about, pseudomonads, you could certainly do phosphate limitation. I mean, the medium in which these guys were evolved was basically a tap minimal medium. It is the case, of course, in any sort of freshwater algal system that phosphate is a limiting nutrient, and so you could have excess phosphate, you could have limiting phosphate, as long as you were able to have growth. It is also possible that there are other nutrients that either favor or disfavor, that is to say, accelerate or retard the propensity of a particular outcome of multicellularity. I think that, so in terms of manipulating the arena, and not just, whether there's, say, a paramecium or a rotifer in it, we could certainly modify the medium itself and see whether there are certain constituents that are favorable or disfavorable, to this evolutionary outcome. Now, so that's my bonehead answer to number one, bearing in mind that there may be, and I should know this, but I don't. I'm still much more into bacteria and to simple yeasts than this, but my students are teaching me a lot about Volvox and Chlamydomonas. So there may be things known in the literature about the production of extracellular matrix in response to certain environmental cues. And like the old saw goes, you can save 3 months at the bench by 3 days in the library. These days, maybe 3 hours in the library. And so, it might be something that we need to investigate further, simply by doing a literature search before we start doing experiments. Now, you asked the second question, and I can't give you a very satisfying answer, but I can give you the beginnings of an answer because I thought this would come up. And so I think you should be able to see now mutations that are common across. So Kim has looked at 4 different lines, 4 different multicellular forms that were isolated from the B2 population. And you may recall from that one slide, actually this slide up here, where you had certain shared mutations. Here's a list of some shared mutations. And what are they? Here they are. You see that about half of them are present in introns. These are whether these mutations have any sort of phenotypic effect is anybody's guess. Are there any smoking guns that I see in this. Well, there are predictive proteins. There's a SNP in this intron of a dual specific. When you see kinase, you think regulation. Both of these are this is intergenic, this is intronic, whether that results in the differential expression of certain kinases and with the downstream effect of having a increased propensity to evolve multicellularity. Tim, I got to tell you at this point, it's anybody's guess. And obviously it would require experiments both restoring the wild type phenotype as well as knockout and overexpression, of these particular genes to see what the phenotypic effect was on the organism. But the point is that we have some candidates to work with, and at this point, since Kim has moved on to she joined a biotech firm in San Diego, and I am pushing her to wrap this up, this paper up. I think that where this paper is going to stop is with the collection of candidate genes. I may tell a couple of just so stories that are just that, and lay out a program in the conclusion as to the way that we intend to approach this moving forward. Does that answer both your questions?
Sure. I was I would knee-jerk react and predict some kind of regulatory mutation might be involved.
Yeah.
One of the easier.
Yeah, and so which one? So there are in some of these I don't have these. These are mutations that are in common across these B2 multis. There are some particular strains that are hit in targets that are easily construed as affecting ECM production, but these are not shared mutations. They are idiosyncratic mutations that are present in particular B2 strains. So this is common in the B2 isolates that she yanked out of this population that were multicellular. There are individuals that have some of those mutations that are actually, if we'd seen that shared across the board, that would have made my job a lot easier. I think that, but it didn't make my job easier. I have.
You got 20 more years of work here.
Yes. That will keep me out of jail, I think.
Thanks, Frank.
Thank you very much, Tim.
Well, it's 5 minutes past 1, so I think we better wrap it up. But thank you so much, Frank, for your time and presenting at our Rough Cut. It was super informative and interesting. Thank you.
Thank you so much. I really appreciate it, Paige. I think really had a good time. This is actually the first time